Okay, so now we're going to talk about ABO antigen in more detail. So what are these ABO antigens and how they are basically determined into like which blood group everyone is having. So ABO antigens, as I said in the previous video, are basically glycolipids. Okay, so glyco means carbohydrates, basically sugar molecules, and lipid means fat molecules. So here you can see there's a ceramide group. Okay, don't worry too much about the ceramide group because we're going to talk about this during our lipid lecture. These ceramide groups are lipids, and these lipids are anchored to the cell membrane, red blood cell membrane. OK, so that's how it's attached to the cell membrane. Now, the ceramide group is connected with a series of sugars. For example, oligosaccharides. It's a few sugar molecules attached to this one. That's why this is called a glycolipid. It's got a lipid part and also sugar part. So now, for example, if this person is O blood group, they will have glucose, galactose, and N-acetyl galactosamine and galactose sugar, and then the terminal sugar will be fucose. OK, so the terminal sugar will be fucose. So these 5 sugar molecules are attached to ceramide and here anchored to the cell membrane. So this person's blood will be considered as a O blood group, or we can call this person having H antigen. OK, we don't normally use this term. We'll just use the term O antigen. But when you come to A blood group, so they will have same ceramide molecules, glucose, galactose, N-acetyl galactosamine, galactose sugar, the fucose molecules, everything exactly same as O blood group. But in addition, they will have additional sugar molecule in the terminal region. OK, so the terminal region will have N-acetyl galactosamine residue, and that will be attached to this galactose. OK, so that's a huge difference between the O blood group people and people with A antigen. So the A antigen people will have 2 sugar molecules at the terminal region. So one is the fucose, very similar to O antigen. Another one is N-acetyl galactosamine. You must remember this name. OK, this is again for a short answer question or for multiple choice questions. It's really important. So this is the molecule that determining that person's blood as a blood group. OK, if somebody got this sugar molecule at the terminal region, in addition to fucose, they will be considered as people with a blood group. OK, what about the next one? So when you come to the B antigen, same ceramide molecule, glucose, galactose, and acetyl galactosamine, and sorry, glucosamine. Sorry, this is N-acetyl glucosamine. This is galactosamine, and then this is galactose, and then in the end they will have fucose sugar, and here just galactose sugar, not N-acetyl galactosamine. OK, try to remember that. So in B antigen, they will have 2 sugar molecules at the terminal. One is fucose, as similar to O antigen, O blood group people, and the other sugar molecule is galactose. So in A blood group, people will have N-acetyl galactosamine. Here in B blood group, people will have a galactose sugar molecule. So that's a huge difference between A and B. So if somebody got these sugar molecules, their blood will be considered as B blood group. OK, in AB blood group, so all of these will be there, but both galactose and N-acetyl galactosamine will be there. OK, that's why they are called AB blood group. So try to remember this. O blood group, only fucose sugar at the terminal end. So therefore, we will not classify this as a or B or AB. So that's called O blood group. And then A antigen, it will have N-acetyl galactosamine. OK, in the terminal region, and that's what is classifying this blood as A blood group. And then in B blood group, they will have a galactose molecule, sugar molecules at the end, and that's what is determining this blood as B blood group. And then in O, both of these molecules will be present. OK. Good. So the next we are moving on to frequency of blood groups in UK. Which blood group is the most abundant one? This is a really important question that many people ask at various occasions. For example, during emergencies, or during surgeries or a trauma, any major accident. So people will be asking, what is your blood group? Has anyone there with this blood group to donate some blood? So it's really important to ensure that we know our own blood group. Okay, so it will be really important to know what blood group we are carrying, because we might get any emergency situation where we need to know our blood group. Okay, so what is the frequency of blood groups in the UK? So O blood group is the most abundant one in the UK. So generally O blood group people are around 48% of people. Okay, particularly if we classify this into O positive and O negative. O positive people are around 35% in the UK. O negative people are 13%. So always the negative people will be very low in abundance compared to positive people. Okay, you need to remember that. And then when you come to A blood group, around 38% people in the UK got A blood group, and particularly A positive. Around 30% people got A positive, and then around 8% people got A negative blood. Okay, so O blood group is the most abundant one, and then the second frequent one is A blood group. And then when you go to B blood group, only 10% people got B blood group. Okay, so particularly B positive, only 8%, and B negative, only 2% people. And then what about AB, which is again very low frequency or very low in abundance? So they are only 4% people. AB positive people around 2%, AB1 around 1%. Okay, don't ask why this is not 4% completely, because they are rounding up to nearest figure here, and therefore totally when you round up everything within the fraction within fractions, then it will come to 100%. So O blood group is the most frequent one. Second most frequent one is the A blood group, then the third one is B, and then the fourth one is AB. And here you can see AB negative is the very rare blood group. Okay, it's really important to understand. AB negative is very rare blood group, even though all the negatives are very rare groups. Okay, so we need to know a little bit about antibodies before we talk about the blood group matching and the compatibility. So what are antibodies? Antibodies are basically immune molecules that recognize the foreign molecules entering into our body. So when we get any foreign pathogens, for example, bacteria inside the body, immune response will kickstart, and the immune system will recognize the foreign molecules, and therefore they will produce an immune molecule called antibody. Okay, these are mainly produced from B lymphocytes. Again, look at the introduction to blood lecture. We talked about this. So this is basically triggering the immune response. So when antibodies are produced, they will be screened against our own self molecules. We talked about that before. And if there are any antibodies produced against our own self molecules present on the surface of our cells, they will be destroyed. Okay, only molecules that are produced against the pathogens or non-self molecules will be circulating there. And the naturally occurring antibodies will circulate in the plasma of subjects who lack the corresponding antigen. So only some antibodies, particularly for these blood groups, for ABO blood groups, the naturally occurring antibodies will be circulating in the body all the time. We will talk about that in more detail little later. So these antibodies are called maternal antibodies. Okay, so normally these antibodies are produced from fetus or immediately after birth, and these antibodies will be circulating in our body all the time, or the memory cells will be circulating to produce these antibodies constantly throughout our life. Okay, so antibodies produced against our self molecules will be destroyed, and produced against non-self molecules will do their job. That's basically binding to the non-self molecule and destroying it or removing it from the system. Naturally, we are producing some antibodies during fetus, when we are in fetus, or after the childbirth, and these antibodies will be circulating in the body all the time. So there are 5 different types of antibodies. Again, you will study these during immunology lecture. And here we are going to talk about 2 important types. One is immunoglobulin G or IgG. Okay, so it will look like this. It's antibodies. They have like a Y-shaped structure, and they will have a light chains and a heavy chains. This is how it will look like. And then the next one is immunoglobulin M or IgM. These antibodies are called pentavalent antibodies. So basically 5 molecules of this type of monomeric antibodies will join together through the chains, the joining chains, and then it will form these pentavalent antibodies. So it's basically 5 Y-shaped molecules joined together using this joining chains and the disulfide linkages. So IgM or important antibodies, because they are the ones mostly acting as maternal antibodies circulating in the body all the time, particularly against the ABO blood groups. OK, so these antibodies pentavalent, so they can bind to multiple cells or multiple non-self molecules at the same time, because each epitope here will bind to one molecule at least. So therefore, they will have a high capacity to bind to non-self molecules. OK, so what sort of maternal antibodies that we are carrying? So I just mentioned here IgM, or the important maternal antibodies that we are carrying in our blood all the time against against the blood group that we don't have. OK, so we'll talk about that in detail here. So if somebody got a blood group in their body naturally, the anti-B antibodies will be circulating. OK, so these antibodies can bind to B antigens. So they will not have any antibodies against A molecules, but they will have antibodies against B molecules. So they have A antigen and therefore they have opposite antibodies in the body, because even if any A antibodies produced, that would have been destroyed. So this is why these people can never take B blood group into their body. They can't receive B blood group. So if they receive B blood group blood, immediately these antibodies will bind to B antigens present on those transfused blood and destroy all those cells. OK, what about in people who got B blood group? So they will carry B antigen, as we discussed before, and at the same time they will carry anti-A antibodies, maternal antibodies. So these antibodies have the capacity to bind to A antigen, not B antigen. This is why B blood group people cannot receive A blood. OK, A group blood, because if they receive this blood, immediately these maternal antibodies circulating in the blood will bind and destroy these blood cells immediately. OK, try to be very clear. A blood group people will carry anti-B antibodies. OK, therefore they cannot receive B blood. And B blood group people will have B antigens and they carry anti-A antibodies, and therefore they cannot receive blood from A positive or A group donors. And what about the AB blood groups? OK, there's a small percentage of people who got AB blood group. So they will not have any of anti-A or anti-B antibodies, because they have both the antigens on the surface of their red cells. OK, both A antigen and B antigen. Therefore, they will not have any antibodies. This is why they can receive any blood. OK, they can receive A blood or B blood because they don't carry any antibodies. OK, what about O? So they will not have A antigen or B antigen. OK, there's no antigens on the surface of these red cells. Only the fucose terminal sugar will be there, but that's not immunogenic. And therefore, these people will carry both anti-A and anti-B antibodies. OK, it's really important that you absolutely understand this slide in this lecture, because this is the really critical one to understand the entire lecture. So O blood group people, because they don't have both the antigens, they will carry antibodies against both. OK, this is why these people can give blood to anyone, but they cannot receive blood from A, B or AB, because they are carrying anti-A and anti-B. So if you give blood from any of these people, these antibodies will bind to those red cells and destroy them. OK, so try to understand this very clearly. So what about RH antigens? So we talked in enough detail about ABO antigens. These are glycolipids, and RH antigen we previously mentioned that it's a protein. In addition to these ABO antigens, This protein is present in every cell membrane, every red blood cells from those people. For example, if someone is positive, for example, A positive, in addition to A antigens, they will also carry this RH antigen. But if someone is A negative, they will not carry this RH antigen. They will only carry the A antigen. OK, what about in B positive? They will carry B antigen. In addition, they will have this RH antigen. In B negative, only B antigen, no RH antigen. AB positive, yeah, they will have both A and B antigens, plus they will have the RH antigen. That's a protein. And then AB negative, no, they will not have RH antigen. O positive, they will have RH antigen, and in O negative, nothing. OK, so in addition to ABO blood grouping, RH blood grouping is really important. Again, there are so many different types. We are particularly talking about RHT because that's the most important one. And that's what is determining someone's blood, whether it's a positive for RH or negative for RH. For example, A positive, A negative. OK, so try to remember this. OK, a little bit more detail. What are these RH antigens and what sort of antibodies that they can generate? OK, you need to remember that we are not carrying any maternal antibodies for RH antigen. We are carrying maternal antibodies, that's IgM molecules, only for AB antigens, not for RH. OK, so RH antigens are transmembrane proteins, so they are anchored within the membrane and they are proteins. OK, that pass through the membrane 12 times. There are a lot of different antigens, but most common one is RHT. And most of the people got a gene that's autosomal recessive. Therefore, most of the people will be positive. Again, try to remember the frequency slide. That's showing that most of the people will have positive blood group and therefore negative blood groups are really rare. So RH antibodies will be only present in plasma if a RH negative person is exposed to RH antigen. OK, as I said right at the beginning, we are not carrying any antibodies against RHT. OK, and for RHT, only IgG molecule will be produced, which is monomeric molecule. It's basically single Y-shaped molecule, not IgM, not pentavalent antibody. OK, and this antibody will be produced only when RH negative people receive RH positive blood. OK, generally RH negative people will not have the RH antigen on their cell membrane. So if we give RH positive blood to them by mistake, immediately the system will start to produce these antibodies. OK, so try to remember here, RH antigen is a protein and we don't have any maternal antibodies against this RH antigen, and they will be produced only upon exposure to RH positive blood in RH negative people. OK, and this type of antibody is called IgG because it's a single Y-shaped molecule. So try to remember that. OK, what will be the consequences of blood group mismatch? OK, we just talked about ABO and we talked about what type of anti-A, anti-B antibodies that everyone will be carrying. And we talked about RH and positive and RH negative and what will happen if you give RH positive to RH negative person. So there will be a group mismatch, blood group mismatch or incompatibility if you give wrong blood group. For example, here. So acute transfusion reaction will be caused by ABO mismatch, and this will happen within few minutes. OK, because we are carrying maternal IgM antibodies, either anti-A or anti-B or both or none. So this will happen instantly when you give blood. Within few minutes it will happen because they are readily available, so they can bind to the newly invading RBCs and then try to destroy them. So here these are group A red cells, so they will have anti-A antigens. And then you are adding anti-A antibodies against them. So these antibodies, pentavalent antibodies, they've been raised against anti-A antigens, and therefore they will bind to them. So as I said before, because they are multivalent or pentavalent, they can bind to multiple molecules. So here, same antibody can bind to 2 or 3 different cells based on the size. So when these antibodies binding to these cells, they will agglutinate. OK, so it's agglutination. It's just a clumping that you can visually see this clumping. So that's why when we do the blood grouping, we can easily see the blood group mismatch. So if they are mismatched, they will bind to each other with these antibodies and then agglutinate or precipitate everything together. And you can see that clearly. If this is happening in a glass slide, when you are doing blood grouping, it will help you to identify what blood group they are. But if this is happening within the body in real life scenario, it will lead to destruction of red blood cells. OK, so this is acute transfusion reaction caused by ABO mismatch, mainly by these antibodies, and it can cause the agglutination. And then after this complex is formed, the complement mediated lysis will happen. OK, complements are important components of the immune system. They will bind to antigen antibody complex and make pores and try to remove them from the body. OK, that's their main role. So when these new red cells are getting lysed, it will release a lot of hemoglobin. OK, that condition is called a hemoglobinuria, and the hemoglobin can get destroyed. Again, we talked about this in previous lecture. When they are getting destroyed, bilirubin level will be increased in the blood. OK, and ultimately it will get increased in the urine and in the stool. So this will be toxic. Increased level of hemoglobin, increased level of bilirubin will be toxic to someone. OK, immediately that can lead to fever, chills, nausea, and clotting within blood vessels, necrosis of kidneys. All those complications will happen. OK, so you need to remember the ABO blood group mismatch is an acute transfusion reaction because we are carrying the IgM antibodies, anti-A or anti-B already in our body. So when this is happening, the newly transfused blood, particularly the red cells, will be bound to these antibodies and the agglutination will happen, and then the lysis will kickstart within few minutes, and that will lead to increased amount of bilirubin production, which will be toxic to our body. Most of the incompatibility during blood transfusions are happening because of the clerical errors and mistaken patient identity. OK, if you are not doing the blood grouping properly when we are taking blood from the donor and recipients, then there will be an error and you will go ahead with the transfusion, and this will cause a lot of problem, basically destruction of the newly transfused cells. OK, thanks here. I will stop this video and we will kickstart the next video. Thank you.